Hi everyone, it's really a pleasure to be here today. One of the reasons why I decided to pursue cancer research in the first place was because my grandmother passed away from lung cancer, and I know that as much as I can make a difference for her, hopefully I can help others who are going through cancer. And recently my grandfather was actually diagnosed with leukemia, so I'm very humbled and happy to be here today. So as was mentioned, I'm a postdoc in Dr. John Bell's lab, and what we primarily focus on is designing viruses to kill cancer cells. So I will also touch on this as well throughout my talk. But first, I'll just give some basic information about cancer cell biology, because really we need to understand this even more nowadays than before to make sure that we're developing the appropriate treatments for patients. And I'm going to take you back a few years. Oh, is it okay? No, it's okay. I won't need it. Thanks. And so cancer is not a modern disease. I'm often asked if it is. And we can see signs from fossils from dinosaurs and mummies that it's been a disease or a curse on our genes for many years, since the beginning of humans probably. And so you can see here from X-ray and high resolution images that mummies showed signs of bone lesions from metastatic prostate cancer here in this case. In fact, one of the oldest documents and papyrus stated that there was a bulging mass, which was breast cancer that they had detected in a patient, but under the therapy for this disease, it was listed there is none. So we've come a long way since then, and I was inspired when I read a book called The Emperor of All Maladies. I'm not sure if some of you have read this book or seen the documentary now, but it talks about a lot of the initial clinical trials that were done with leukemia patients, especially childhood cancers. And I think that's a big example of all the success we've had recently, and it's laid out the ground for some of the research that's being done today. So cancer is a collection of over 200 diseases. Obviously, they will require different treatments and different cures. I don't need to probably talk about all the statistics, which I know we're all too familiar with, but I will mention that, as Robert Weinberg said, if we live long enough, sooner or later we would all get cancer. As we know, cancer is caused by mutations over time, and if we live longer, there's more chance that we'll get cancer, especially if we outlive other diseases, as we seem to do in Canada. So genes control all aspects of growth, from early hours to weeks of our development and decades and beyond. And mutations in single cells are what give rise to cancer. So when we have normal cells, they behave normally, but when there's DNA damage, this leads to mutations and therefore the uncontrolled cell growth, which develops into a tumor. So here on the left side you can see is what would be a monoclonal tumor, which might be easier to treat than, for example, on the right-hand side, a polyclonal tumor. And this is what some of my colleagues were referring to when they say that cancer is heterogeneous. It just means that it's made from many different populations within it and could make it more difficult to treat. What genes are mutated in cancer? So specifically, they tend to be classed into two groups. So proto-oncogenes, which tend to be activated. Their normal functions are in controlling cell proliferation, migration, survival. But if you have an activating mutation in these proteins, it tends to not allow them to be shut off, so they're always on. There are also tumor suppressors, and these are genes that are important for DNA repair. And if they are inactivated by a mutation, then you no longer have the brakes, you could say. And the biggest example is p53, which is known as the guardian of the genome, not the galaxy. And so you've heard today that p53 is a big player. And so this is a list here of some of the different driver mutations in CLL specifically. And you can see towards the top, you can see p53 is there alongside some of the other deletions that were mentioned and other novel players that are being teased apart at the moment. This is a publication from Nature from just 2015. So you can see a lot of the underlying causes of CLL are still being studied to develop new therapies. I'm also often asked, what are some of the causes of cancer? Because if we can predict what they could be, maybe we can prevent some of them. And so a small proportion, probably about 10%, is linked to hereditary familial cancers. And usually there's screening for different types of these cancers. The biggest example, for example, was probably the Angelina effect recently, where she knew she had a BRCA1 mutation and it would lead her to increased likelihood of developing breast cancer, and so she opted for a double mastectomy. There are also immune deficiency syndromes. And so we heard today, for example, that with CLL there is a bit of a hereditary component, but I don't think those features are really clear yet, and they're still being explored. But it probably is a good reason to also to know your mutational status, because it's important not only for yourself, but your other family members if they might end up having a similar disease. So some causes of cancer that are environmental. I'm sure you're familiar with UV and ionizing radiation. And you can talk to your physicians about this, but checking yourself out for looking at differences on your skin is important, especially as we talked about secondary malignancies leading to skin cancers. And in events like Fukushima, for example, where there's ionizing radiation tends to lead in increases in thyroid cancer, for example. There are chemical carcinogens, so tobacco smoke being the leading cause of cancer. There's also asbestos, benzene, aflatoxin, arsenic. Radon gas is the second leading cause of lung cancer. It's found in your basement. Most people can buy a kit at a home hardware store to test for it. Many Canadians don't. It's something that I advocate. Interestingly, when I was doing some reading about CLL, there seems to be a weak association, maybe with Agent Orange, which was a mixture of herbicides used in the Vietnam War. Alcohol use, unhealthy diet, physical activity are other reasons. Some other causes that I think people are less aware about are biological carcinogens. So certain bacteria like H. pylori can lead to gastric cancers. There are some parasites, like this one, that can lead to bladder cancer. And there are viruses, HBV, HCV can lead to liver cancers. And there's also HPV, and nowadays that's why we have vaccines in schools for both boys and girls. It doesn't only cause cervical cancer, but as shown in this pyramid here, it causes a variety of other types. And we can prevent those with some of those strains with a vaccine, and HIV. So just to, again, summarize some of the aspects of cancer prevention, I think eating healthy diets higher in fiber and greens is important, less red meat. It's been shown to be correlated to less likelihood of developing colorectal cancers and other types of cancers too. Exercise, sunscreen, vaccines. And I would also like to put a warning also in patients who take supplements that aren't necessarily regulated. If you do take anything, I think it's important to also tell your doctor what you are taking, especially when you're on some of these drugs. So, cancer therapies. So you've heard about different forms of therapies such as surgery, radiation, and chemotherapy, and some novel targeted therapies that are coming into play now. But what I'm really going to focus on is immunotherapy, specifically oncolytic viruses and CAR T therapy. And I think with all of these, some of the challenges include resistance and toxicity. On the right-hand side here, just as a reminder, is your defense system in your body. So you have NK cells, macrophages, dendritic cells. The focus is today, I think, has been mostly on your B cells, obviously, and how we can harness the power of T cells. So as I get into oncolytic viruses, I'll just say that on the left-hand side here is a summary of hallmarks of cancer. So this was a famous publication in 2000, 2011 as an updated version that talked about the different characteristics of cancer cells. So some of these include how cancer cells can avoid, resist cell death. They can invade, metastasize, which is what really contributes to patient death. It can lead to tumor promoting inflammation, avoiding growth suppressors, sustaining proliferation, so continued cell growth, which leads to those tumors being formed. But what I want to drive a point here on is the fact that oncolytic viruses, so viruses that naturally or have been engineered to only replicate in cancer cells and leave normal cells unharmed, they actually exploit these same mechanisms in cancer cells to do well. So the same genes that control cell growth and death also control antiviral programs, and we can harness this and take advantage of it. So our bodies are made of billions and billions of cells, and viruses are parasites that can only reproduce inside of those cells. So we've basically designed in our lab viruses to be parasites of cancer cells. And viruses not only lyse through a process called oncolysis, the cancer cells, but what we find is even more important is their ability to alert the immune system once again that, hey, this is a cancer cell you forgot. So we have different models of metastatic cancer in our lab where we can inject cancer cells in the mouse tail vein. A tumor will form. As you can see here, it looks kind of nasty on the right-hand side. Poor little mouse. When we treat the virus, though, 14 days later, we could see that the tumor actually clears and the lung looks unaffected and back to normal again. So mouse experiments are important. They can lead to therapies for patients. It's an important first step before we can test things out in humans. And this is an example here where it was an intravenous delivery of a virus, and it allowed for the replication in the cancer patient. And you can see that there's evolving necrotic tissues where the virus was able to invade. But we need to maybe, sorry, I'll touch on that afterwards. So tumor responses after following oncolytic viruses. So these are some examples here of phase 1, phase 2 trials where you can see that the results might only be happening in a small percentage of people at first. However, we've gone on to take a look at what else can be beneficial in combination with some of these viruses and how we can enhance the power of these viruses. So here's an example shown when we implant a colon tumor in a mouse. We administer an oncolytic virus. We're able to cure the mouse, and we can re-inject a tumor in the mouse, and we see that the mouse actually rejects the colon tumor. So the immune system can still detect the tumor and doesn't allow it to grow in the mouse once again. Same thing here in humans. Viruses kill tumors, but the immune system keeps them from coming back when we look at several days past the time by which when they were treated. Sorry for some of these graphic images. So in this case here, the oncolytic virus was administered in combination with an immune checkpoint inhibitor. I'm sure you might have heard of anti-PD-L1 or PD-1 or CTLA4. These have been very popular. And here we've been able to show that there's a great deal of success in combining these with oncolytic viruses. So cancer's like a fire that continues to spread, and what we really need with our oncolytic viruses is to target many different aspects of it. So looking at immune cells, shutting off tumor blood supply, tumor targeting antibodies. We want a virus infection and cell lysis as well. So the combination of this can help a lot. But more recently, there's another avenue that we can combine oncolytic viruses with, and that includes immune cell therapy. So immune therapy is a new class of cancer treatment designed to boost or enhance the body's immune system to more efficiently target and destroy cancer cells. Here's an image of a cancer cell getting attacked by 3 T cells, which is pretty neat. And so this is an image that really depicts the power of immune cell therapy. And here's a bit of a case study. Emily Whitehead, she was a patient who had ALL at 5 years old. She was enrolled and was the first pediatric patient to receive genetically modified T cells to fight leukemia. And shown here a picture of her in 2017 is her 5 years cancer-free afterwards, which is pretty amazing. So CAR T cell therapy, as a reminder, it's a lab-made protein, a CAR protein that's added to the patient's own T cells. And this CAR protein helps T cells target cancer cells to be killed. Most of the trials at first were with patients who had ALL. And so this led to the FDA approval of CAR therapy just last year, 2017, for this. However, there are ongoing clinical trials to test out the efficacy of CAR T therapy in CLL patients as well. And so while the response rate is a bit lower for CLL patients, some have remained in remission since treatment, and this would be called a durable response. And there are several new generation of CAR T therapies in development too that are showing more promising results. So you've heard earlier about CD19. This is the ideal target for CAR T cells because it's a cell surface protein found on the B cells and on B cell precursors. Importantly, it's not expressed on the hematopoietic stem cells, but it's expressed on most of the B cell malignancies, including CLL, shown here on this diagram. And you can see the wide expression of CD19 on these different malignancies, or cells. And so CAR consists of T cell activation domains, shown here extracellularly and on the outside here at the top. And it's coupled to an anti-CD19 single chain variable fragment. And within intracellularly, what you can see here are domains. So the extracellular domain recognizes CD19 on the malignant cell, and the intracellular portion of it is important for the activation of the T cell. And so some of these are some of the first clinical trials that took a look at designing these CD19 CAR T cells. I think the most successful and the very first was through Novartis. A question that's asked oftentimes is what's the cost of this? I think for Novartis, CAR T therapy costs $450,000 per patient. And with other medical expenses, it can go up to $1.5 million. So I think this raises the question, what happens after the clinical trials? Who will cover this? Feel lucky that in Canada, hopefully the medical system will be able to cover most of it. And in the US, I guess a lot of the insurance companies, you'll have to look into that to see what they can do. So as I said, things are still in its infancy. Clinical trials for this are still recruiting. At the moment, I think there are about 240 different trials on the go. So it's very promising, and I think it's an area that people need to look into more. So again, just to reiterate what happens, blood samples collected, they extract the T cells, and then in vitro, in petri dishes in the lab, they will use a virus, which plays an important role, to transduce the cells and put this CAR protein on the surface of these T cells. Then we allow for expansion of the CAR T cells, and then they're administered back to the patient. There are two different approaches for CAR T cell therapy development. One is allogeneic, when they'll engineer T cells from a single donor and then utilize it for multiple patients. This might actually end up being a more cost-effective way of doing things in the future. But there's also autologous CAR T cells that are engineered for every single patient from their own T cells. In 2015, there was a publication in Science Translational Medicine, and they talked about how CAR T cell, they persist and induce sustained remissions in relapsed refractory CLL patients. Of note, they showed 4 complete remissions and 4 partial remissions, although all responding patients developed B-cell aplasia and experienced cytokine release syndrome. So some of the side effects of CAR T-cell therapy, they were briefly touched on earlier, but the major one is cytokine release syndrome, I would think. It correlates with T-cell proliferation and efficacy, which shows that the therapy is working, but as Dr. Kipp said, it's kind of like an avalanche. There's just too much happening with too many cytokines being released, and it's definitely a stress on the body that can lead to patient death, unfortunately. So some of the cytokine release syndrome, they've noticed that it leads to increased levels of IL-6. So they're now developing cytokine-directed therapies that can help to alleviate cytokine release syndrome in patients. There are also some neurotoxicity, tumor lysis syndrome, and loss of normal B cells that are associated. And any patients that are interested in joining CAR T-cell therapies will be told specifically for them if it, what side effects they would expect and if it would work for them or not. So future directions, building a better CAR T cell. So I think ways to, as was mentioned, learn more about the tumor microenvironment to make sure that where we can target all of the cancer cells is very important. The successes of CAR T have been seen in ALL patients and CLL patients, but hasn't been the case yet for solid tumors. So I think a lot more work will be done in this area. CAR T trials are starting to target other antigens like CD22, ROR1, and others that have been mentioned earlier today. And I think people are starting to look at also the combination of ibrutinib and other drugs with CAR T. So immune cell therapy, it's a game-changing technology, but I think it works in a limited number of cancers. Luckily for you, it seems like people are starting to focus on ALL and CLL and the benefits there first. But I think that understanding the side effects and trying to generate better CAR T cells are very important. And our strategy in our lab will be to combine oncolytic virus attack on the tumor in combination with CAR T cell therapy. If we look at regional disparities in the studies of CAR T cell therapies, we can see here that North America, Europe, and China are ahead of Canada. But we're still leading the way, and it's still very early on. This is a publication in Science just this year. So I think we have a lot to do still in Canada. Luckily, Dr. John Bell and other collaborators have put together through BioCanRx and partnerships with BC Cancer Agency and the Ottawa Regional Cancer Foundation, a plan to look at blood cancers and design CD19 CAR T therapy for those who are relapsed or refractory with their B-cell malignancies. And so there's a clinical trial that's expected to start early 2019. And clinical trials are a great opportunity to learn a lot more than we can from mouse models and other work in vitro, but all of it together, basic cancer research and translational, is important as a whole to develop to make new discoveries and to further the work and to see the effects in patients that it can have. These are some other clinical trials that are planned for the next 2 years at the Ottawa Hospital Research Institute. So once again, at the Ottawa Hospital Research Institute, we have different labs that are focused in different areas, so manufacturing some of the viruses that we use, testing them in patients, and discovery, as I mentioned. So lastly, I'd like to thank everybody. Here's a picture of Dr. John Bell in the bottom left corner. He's pretty happy to have received just recently the Golden Virus Award at the International Oncolytic Virus Conference. And the rest of our team, who I must thank for all of their contributions as well to make our institute what it is today. Thank you.
Fantastic. Thanks so much. We do have time for some questions, I believe. Yeah, we're good. So I think if there are obviously technical questions, we'll direct them to Mathieu. If there are clinical questions, we can always try and tackle those as well.
You asked good questions, but I'm going to come back to this lady here who has not asked any question until now.
There you go. I was curious about the genes you mentioned early on, or the cancer-specific genes. I know the breast cancer is the BR, but are any of those others available to be tested? Is it something you just have found, or can you be tested for those genes?
So generally, I think physicians will tell you if you need to be tested for any of the different genes associated with cancer, most likely if you have family history, for example. But I think those genes, some of them are a lot more clear for some of the other cancer types than they are maybe for CLL.
You mentioned, sorry, over here.
Yes.
You mentioned that you're going to learn more from one human clinic patient than you could from all the mice. However, I would temper the enthusiasm with the fact that a lot of these lessons may be very hard ones, and that we, the guinea pigs, so to speak, are being asked to throw the dice with a lot on the table. I hope you bear this in mind when these trials begin.
Certainly. And on that matter, though, a lot of the phase 1 clinical trials are patients who have exhausted all other forms of treatment. Not to say that it means that we need to not care about the patient. We definitely do. Doses are tailored for the patients, and the physicians are there to monitor the progress of these different drugs.
Hello, doctor. I was wondering, I had a similar question to his. I was wondering how you reassure patients that the virus, number one, is exclusive to the attacking the cancer cell, and what happens to it long term. I mean, viruses get in and they have a habit of hanging on and replicating. And so my question is similar, and my concerns are because earlier in the history of oncolytic virus therapy, there were some tragedies, and this was probably a good 10 years ago. Along with your talk, I would like to say to folks that I posted this morning, which would be a nice follow-up to this, that OSU is having an informational live stream program on clinical trials. And if you go to your favorite forum, I've just posted to all of them clinical trial information, and you can register for free. So I just wanted to, say that this would be a good follow-up for this, and thank you for your presentation. Oh, one other thing. I want to get one other thing in. The thing is, do you ever use, do you ever consider using CRISPR-Cas9 with your techniques?
Okay. So to first answer your first question, in 2015, the FDA approved the first oncolytic virus for its use, which is TVEC for melanoma, and it has had tremendous success. And I think this is great, actually. In fact, if there isn't secondary malignancies of melanoma in CLL patients, that might be an opportunity there to have a certain type of treatment that would work. And these other viruses have undergone already through phase 1 clinical trial. They are safe. Patients are not dying from administration of these viruses. And we are seeing successes, as some of the pictures I showed. CRISPR-Cas9, some different companies are starting to use it in how they're designing some of their strategies for targeting cancer. But again, I think it's also in its infancy. Thanks.
Dr. Crupi, I know that your team in Ottawa is working towards a CAR T trial, and that as the lead up to the trial, you are developing a protocol for how to structure the trial and how to manage it. And we heard that you're also looking for patients who might want to provide input from a patient perspective on how the CAR T trial should proceed. So if there's any Canadian participants in the forum who are interested in participating in that, you should probably let Dr. Crupi know, or myself. We're going to post something on a CLL PAG website, but this is a good thing when researchers are asking patients for their points of view as they develop clinical trials. And so your team is commended for taking that approach. And if you are interested, certainly let us know.
Certainly understanding what's important to patients about how they're going to be enrolling into these different clinical trials is something that's really important to us. And the lead on that is Madison Foster, who I can put anybody in touch with afterwards. Yes.
Question. If somebody has a CLL type of cancer and you develop a virus, to attack it, doesn't that virus have to be programmed specifically to each individual? Are you saying earlier that you're growing viruses that will be able to work with anybody or a larger group of CLL patients?
These viruses could work in anybody, yes. So that's one thing that's an asset to it. We can tailor them for specific types of cancers. You can imagine an oncolytic virus being a battleship. So there's a lot of room within the virus so that we can encode even antibodies within it to be secreted in the microenvironment.
Thank you.